Okay guys, in this video I'm going to go over cell respiration for anybody that doesn't understand it and needs to know for the test tomorrow. Okay, so there are three steps to respiration. The first step is glycolysis, the second step is the Krebs cycle, and the third step is the electron transport chain. The link step is the step between the glycolysis and the Krebs cycle, but only occurs if you're going through aerobic respiration. So, step 1, glycolysis produces, as you can see here, 2 ATP and 2 pyruvate. So, step 1, glycolysis. You start with a carbon molecule, which you see over here, which is glucose, 6-carbon molecule. The glucose undergoes phosphorylation, where 2 phosphates from the 2 ATP are turned into ATP. Both those ATP donate an inorganic phosphate. And those inorganic phosphates are stuck onto the end of each one of the end carbons in the 6-carbon molecule. This one and this one. Okay? So, then that molecule is called fructose 1,6-bisphosphate. And that fructose 1,6-bisphosphate is the first step towards getting the pyruvate that you want at the end of glycolysis. This fructose 1,6-bisphosphate is a 6-carbon sugar. Then it goes through a step that is called lysis. Lysis is when the fructose 1,6-bisphosphate breaks into 2 G3Ps, which is also the same product of the Calvin cycle in photosynthesis. And as you know, that G3P in photosynthesis is used to either regenerate RuBP or it's used as a byproduct, which is very diverse throughout the cell. Now, these G3Ps, there are 2 of them, as you can see right here. Now, it looks like this. And these are called glyceraldehyde 3-phosphates, not glycerol. Glyceraldehyde. Okay? So, that's lysis. Then you go through a step called oxidation, where you have the 2 inorganic phosphates added to the G3P. Now, how does this happen? What happens is that you have NAD plus come in, which is a electron carrier, and the NADP and the NAD plus is reduced to NADH, and that happens because it is picking up electrons from this step and turning into NADH. Now, from that change, there's a little bit of energy released, and that energy is used to combine the 2 inorganic phosphates to the G3P. So you're left with a molecule that looks like this through oxidation. Now, and you have 2 of those. Okay? And so you also have here in the next step, which is just a step after that, there's no name. It's just used to convert the G3P with the inorganic phosphate into pyruvate. You have 2 ADP which come in, and they take the 2 inorganic phosphates to 2 ATP, and they turn into 2 ATP. Now this happens for each 1 G3P with the inorganic phosphate, so it happens twice. That's why there's the 2x over here. So here you have a net gain of 4 ATP. And because of that, you lose 2 over here, and you gain 4 over here, so you have minus 2 plus 4, so you have a net gain of 2 ATP, which is what's one of the products from glycolysis. And after that, you're left with a 3-carbon sugar without the inorganic phosphates, and it is called pyruvate, and you have 2 of them. And this pyruvate is what's used to go through the link step in aerobic respiration, which is the focus right now. So these pyruvates, what's going to happen in the link step is this, okay? So first, I just forgot to mention that the pyruvate, the glycolysis occurs in the cytoplasm of a cell. Now, pyruvate occurs across membranes between the cytoplasm and the inner mitochondrial matrix. So in pyruvate, what you're going to do is, first of all, pyruvate only occurs in the presence of oxygen. So you need to know that if there's no oxygen present, then the pyruvates from the last one will go through the anaerobic respiration, which is when you go through either fermentation to produce either ethanol or lactic acid. So in the link step, you have 2 pyruvates go through the inner membranes into the mitochondrial matrix, and this is just the gist of it. What happens is you're basically oxidizing the acetyl group in the pyruvate to produce 2-acetyl-CoA. Now, these 2 pyruvates undergo active transport to turn into this. What happens is described in more detail here. This is the pyruvate. What's happening is that this carboxyl group in the pyruvate is decarboxylated, so it leaves. And it leaves in the form of CO2 when it's going through active transport to enter the intermembrane of the mitochondria. So, there are 2 pyruvates. It's going through twice. So you have 2 CO2 released as a byproduct. And so while this is happening, you have NAD plus two NAD plus coming in and becoming reduced, which is basically picking up electrons and turning into 2 NADH. And those NADH are electron carriers. And they take electrons out. And what else is happening is you have coenzyme A coming in to help facilitate the reaction that turns pyruvate into acetyl-CoA, which is expressed here. And this, remember, you need to know that this is active transport. And active transport goes from a low concentration to a high concentration. It requires energy. However, in the link step, you need to know that no ATP is used and no ATP is produced. So, this is explained more here. Coenzyme A. Coenzyme A helps form the pyruvate to acetyl-CoA right here. And CO2 is a byproduct, and NAD plus to NADH is an electron carrier that picks up electrons and takes it to the electron transport chain. To the Krebs cycle, actually, sorry. And the big things you need to remember from the link step is that pyruvate is oxidized to acetyl-CoA and NAD plus is reduced to NADH. So your products will be 2CO2, which is the byproduct, which is right here, and the 2-acetyl-CoA, which is shown here. And as you need to know that acetyl-CoA is a 2-carbon sugar. That's really important. Okay, now what happens is that this acetyl-CoA goes on to the Krebs cycle. Okay, the Krebs cycle is also called the citric acid cycle, and it's only going to happen if ATP levels are low. So if ATP levels are low, then the inner mitochondrial matrix is going to have the Krebs cycle. So what happens is you have your 2-carbon sugar, acetyl-CoA, and this is just, by the way, showing for 1 acetyl-CoA, not 2. Your acetyl-CoA combines with OAA, which is a 4-carbon sugar, to produce citric acid, which is a 6-carbon sugar. Now, this coenzyme A, which is from acetyl-CoA, this CoA is recycled and used again in the link step because it's an enzyme, and as we all know, enzymes can be used multiple times. So, you have citric acid, which is produced by the combination of acetyl-CoA and OAA. So you have citric acid. Then you're going to go to the next step from a 6-carbon sugar to a 5-carbon sugar. And in the process, you're going to release 1 CO2. It's going to be decarboxylated. And you're going to go from NAD plus to NADH. So NAD plus is going to come in and pick up electrons, hydrogen ions, to go to NADH. Okay? So here, the 5-carbon sugar is going to turn to a 4-carbon sugar. And the same thing that happened previously is going to happen again. The CO2 is going to become decarboxylated and leave as a byproduct. And NAD plus will come in and reduce to NADH. Now both these NADH and the one up here, which I'll explain later, will go to the electron transport chain to participate in the production of more ATP. So then to turn from a 4-carbon sugar back to OAA, you're going to go through multiple steps. First, ADP is going to come in and dephosphorylate, and it's going to pick up inorganic phosphates and turn into ATP. And another thing that's going to happen is you're going to have another electron carrier called FAD plus come in and take electrons and turn into FADH2. It's going to be reduced again, just like the NADH is up here. And speaking of NADH, this NADH is going to be reduced again, just like this one was here. So what's finally going to happen is you're going to turn this 4-carbon sugar into another 4-carbon sugar, the OAA. And that OAA will be used to combine with the next acetyl-CoA that comes in. So through the Krebs cycle, you have a product. These are your products. For 1 acetyl-CoA, you have 2 CO2 produced, 1, 2. You're going to have 3 NADH produced, 1 here, and 1 here. You're going to have 1 FADH2 produced from right here. And you're going to have 1 ATP produced right here. So because you have 2 pyruvates, which turn into 2 acetyl-CoAs, for 2 acetyl-CoAs that come in, this is going to be the products. You basically take these and multiply them by 2. Okay, so then what's going to happen is these products are going to be good, but these NADH and this FADH2 is going to go into the electron transport chain, which is used to produce multiple ATP. So what happens is, and I forgot to mention, the Krebs cycle occurs in the mitochondrial membrane matrix. Okay, and so then you have the electron transport chain, which is the location is the intermembrane space of the mitochondrion along with the intermembrane of the mitochondria. So, from the Krebs cycle, you have NADH2 and FADH2, which are electron carriers. Now, what they're going to do is they're going to take the electrons to either NADH dehydrogenase, that's where the NADH2 takes them, or FADH2 will take it to the ubiquinone, which I just abbreviate as Q, and that's what you're supposed to do. So, what's going to happen is, one thing that's worth noting is that NADH dehydrogenase occurs at higher energy levels than FADH2 does. So this is going to result in a production of 3 ATP, while this will only work in a production of 2. So NADH hydrogenase is going to take 2 electrons and rise them to a higher energy level. And in the process, the chemiosmosis is going to occur, and hydrogen ions are going to be released into the intermembrane space of the mitochondria, which is the space between the 2 membranes. Then, the 2 electrons that were raised at a higher energy level will go to the ubiquinone. The ubiquinone is just an electron carrier. The ubiquinone will take it to cytochrome BC1, which is a protein complex that holds electrons, and here, other chemiosmosis will occur again, and it's going to be release another hydrogen ion. Okay, so into the intermembrane space, which we use later. It's important knowing that this chemiosmosis is known as oxidative chemiosmosis, and that is a type of chemiosmosis that occurs during the lowering of energy levels, like when, in example, an electron transport chain. Furthermore, you're going to take the cytochrome BC1, and the electrons inside are going to go to the cytochrome C complex. And it's important knowing that the cytochrome C complex can only take 1 electron at a time. It cannot take 2. So what the cytochrome C complex is going to do is it's going to take 1 electron at a time, and it's going to drop them off at the protein complex called cytochrome oxidase. Now, what the cytochrome oxidase does is it just keeps dropping off electrons. And you have O2 just waiting here. Now, this O2 is highly here, let me draw it better. It's kind of hard to see. This O2 is highly electronegative, so it's going to pick up the electrons. Okay? But see all these hydrogen ions that were occurred outside? What's going to happen is there's also going to be a buildup of hydrogen ions inside. And what they're going to do is they're going to go through the cytochrome oxidase, and they're going to diffuse outward. This is another chemiosmosis. But what's going to happen is some of the hydrogen ions are going to bind with the O2 along with the binding of the electrons. You have 2 hydrogen ions that bind for every electron, so that's going to produce H2O, which you can see over here. And so that's how water is produced through the process of cellular respiration. And so what's going to happen to the other hydrogen ions is that they're going to diffuse out into the intermembrane space through chemiosmosis, just as they did here and here. So what's going to happen is you're going to have a hydrogen buildup in the intermembrane space. And because you have a buildup, right, like it's shown here, these are going to want to go from an area of higher concentration to an area of lower concentration. They're going to want to diffuse into the matrix. And some of them, although some of them go through the membrane, most of them are going to go into the membrane using ATP synthase. 3 hydrogens go in at a time, and they go into the membrane. But what also happens is they serve as the energy which pumps ADP, which pumps them through, and they also cause ADP to bind with an inorganic phosphate to produce ATP. This is going to occur multiple times. That's what you're going to result in is you're going to result in 32 ATP as a grand total. So what you generate from this entire cycle is you're going to generate 6 water molecules and 32 ATP. So from every step in cellular respiration, I'm going to go through that right now. Glycolysis, you produce the 2 ATP from the net gain of 4, and you produce the 2 pyruvates. From the Krebs cycle, you produce the 2 CO2, as you can see here, 1, 2, as byproducts through decarboxylation, and you're going to produce 2 FADH2, because 2 acetyl-CoAs go in, and you're going to produce 6 NADH plus H+. Okay, plus 3 for 1, so 6 for 2. And you're also going to produce 1 ATP for each acetyl-CoA, so you're going to produce 2 in all, because there's 2 acetyl-CoAs. In the link step, which I actually skipped, you're going to produce 2 CO2 through the decarboxylation of this carboxyl group, 2 of them, because there's 2 pyruvates, and you're going to produce 2 acetyl-CoAs with the help of coenzyme A. And through the electron transport chain, you're going to produce 6 water molecules because of this step right here, where the O2s bind to the electrons and the hydrogen ions and produce H2O. And you're going to produce 2 CO2. You're going to produce 2 CO2. And you're going to produce 32 ATP. This says 34. It should be 32. So now, totals, what you're going to get is you're going to get your 2 CO2 from here, your 2 CO2 from here, and your 2 CO2 from here. Right? Actually, I made a mistake. You're going to produce 2 CO2 in the link step. You're going to produce 4 CO2 in the Krebs cycle because you produce 2 for each acetyl-CoA, so 2 times 2 is 4. And you're not going to produce any in the electron transport chain. And another thing that's going to happen is that you're going to produce your 32 ATP from the ETC, your 2 ATP from the glycolysis, and your 2 ATP from the Krebs cycle. So you have 32 plus 2 plus 2, so a total of 36 ATP. So your total products of cellular respiration, aerobic cellular respiration, are 6 H2O, 36 ATP, and 6 CO2. So that's cellular respiration. Sorry for my continuous mess ups, but I hope that helped. Cool. Bye.